Yeah, let's talk about the mitochondriopathies. So there were very few, and um, all you knew was they were syndromic and you couldn't do much about it. However, now we know that there are secondary causes as well. And this becomes very important because that means you and I can have some effect on some of the patients that get affected um, by mitochondrial dysfunction. So it was exciting. About 3 weeks ago, we did a podcast and I interviewed the scientists that cracked this test. So it's the University of San Diego in the US, California, and Dr. Patel's team basically cracked it. This has been his life's work. His interest has always been in mitochondrial function. And as a junior doctor, this is what he worked on, and then moved on to lab medicine and eventually came back to it. And it's exciting because now, without doing a biopsy, which is what we used to traditionally do in terms of working out how the mitochondria is functioning, now you can do a blood test and you're able to ascertain the function of mitochondrial function. So this test is now available in South Africa, and the company is ME Screen, so capital letter M, capital letter E, and Screen. And you can order these test kits and then obviously sell it to your clients, or your clients can go online and order it themselves, and then the report can be sent to the treating physician, and then you can try and manage these conditions. So today it's we managed to get you guys CPD points, 2 for today, because we're going to do 2 modules today. And then more importantly, is going to be the second 2 modules, because there you'll able to see how you can have an effect on treating some of the symptoms of mitochondriopathies. So today we're just setting the background. We've got people from varied backgrounds, so it's important to go through some of the basics. So module 1, we're going to look at how the mitochondria works and their relevance to health, wellness, and disease. And we're going to tackle module 2 today as well, where we're just going to look at the basic physiology and biochemistry of the mitochondria. So what are the learning objectives of this program? To understand the broad role that the mitochondria plays in the physiological processes in the body and their relevance and the connection to health and then disease. You must be able to identify the core biochemical processes involved with the mitochondrial function by the end of these 4 modules. We must be able to differentiate current testing approaches used to assess function, understand the limitations of testing approaches in determining primary and multifactorial mitochondrial disease and dysfunction, and then review the application of the functional markers that you will pick up on your tests and assess the efficiency of the mitochondria and their function. So let's go through the first one, and it's a basic one, basically going to first year university, either BSc biology or medicine. So mitochondria, their role in health, wellness, and longevity. Here we're going to review the role of the mitochondria in human body, understand the basic structure of the mitochondria, describe how mitochondria are linked to health and how it's relevant to certain disease processes. Now, this is important because if you go and watch some YouTube videos on mitochondria, it's become huge in the functional medicine space, in the homeopathy space, and it's moving into mainstream medicine as well. You'll see that there's about 12 products that can be used to have an influence on the mitochondria. Now, historically, you couldn't test for it. And so if you can't test for it, you can't have a baseline. And that means which one of the 12 products are you going to use? And if you choose 1 or 2, how do you assess whether you've achieved what you want to? Let me put this in another way. Traditionally, with patients with type 2 diabetes, you know they've got diabetes because they've got the symptoms, and you've got a confirmatory test where the sugar levels are up or the HbA1c levels are very high, and you will then intervene. And then 3 months later, you've got a test for yourself and for the patient and for the funder to see whether the condition has improved or not. So in medicine, that becomes a very important role to have a control. Yes?
Excuse me, could you possibly enlarge your screen? We can only see a very small, the screen is very, very small.
So I'm going to try, but I started by saying that unfortunately we couldn't modify this PDF because it comes straight from the University of in California of San Diego, and we tried to get them to send us a format so that we could put it into the usual sides that we do. So when I tried to make it a bit larger earlier, some of the third or the fourth slide just cut off. Can you see what's happening here now? And that means we're going to have to do the rolling action. And some people find it difficult. Like if you look at this slide now, the entire right side is cut off. So I apologize. And I know the writing is small, and maybe we'll share the slides. We'll ask the company if we're allowed to share the slides, and it should be fine. So unfortunately, I am well aware. And today we've been trying to get them to send us a format that we could use in South Africa. And in fact, I had prepared some of my own slides in between to make it a bit more interesting and relevant to us and take us back to a bit of first year biochemistry. But let's see how far we can get with this. So,
Doc, what if you close that box that says format on the right-hand side? That might actually open up the frame.
Okay. Better?
Yeah, much better.
I needed you earlier today.
And then you can enlarge it even more at the bottom right-hand corner where you were doing it before. There you go.
So I'm going to still have to do the rolling thing. I hope you guys don't.
There you go. Perfect.
Okay. So mitochondria are organelles within the eukaryotic cell, right? And they produce basically ATP, which is a primary energy source all living organisms use. So we think that mitochondria only produce ATP, and that's the main function in terms of cellular respiration. They do it mainly through aerobic respiration, and they require oxygen. However, if you look at the bottom part here, you'll see that the mitochondria have many other functions outside of energy production that we haven't concentrated too much on in the past. It's vitally important for calcium storage and buffering, very important for controlling cell death and survival, so apoptosis, and you basically need to kill old cells and allow new ones to be produced. Otherwise, it accelerates the aging process. We need to they involve in regulating cell signaling, and then very important for thermogenesis. And we're going to try and cover those in the 4 modules. So the number of mitochondria in the cell depends on how much energy that particular cell requires. So obviously muscle cells are going to have a lot more mitochondria because they need to produce a lot more energy. However, you'll also note that the mitochondria in the muscle of Usain Bolt is going to be different to me and to Bruce Fordyce. And Bruce Fordyce is probably going to have the most mitochondria because for long distance running, you're going to be using more aerobic respiration, whereas Usain Bolt is going to be more anaerobic. And so from that perspective, even from individual to individual, there will be a difference. Red blood cells, just out of interest, are the only cells in the body that do not have mitochondria. And remember, they're the ones that carry oxygen around. So let's start by looking at the evolution of how we ended up having mitochondria in us. So if you look at the ancestral prokaryotic cell, so from a very simple life, simple molecules, it got incorporated within a cell membrane. Over millions of years, you ended up having prokaryotic cells. Now, prokaryotic cells behave like amoeba, and that means they have the capability of engulfing. That's how they actually get their food. So by engulfing aerobic heterophilic prokaryotes that were smaller, we ended up having prokaryotic cells that incorporated what we now call mitochondria. And that then became the ancestor to the eukaryotic cell that you and I are made out of. Interestingly, the same process also incorporated photosynthetic prokaryotic cells, and that is what you will find in plants, and that's why they can generate energy from the sun. So we get our ATP production from the mitochondria, having oxygen and various pathways, which we're going to talk about, and then the plants do it through the sun. So from an evolutionary perspective, this is a symbiotic relationship between two organisms, and basically it served each other well from a survival perspective. Now let's look at the structure of the mitochondria. So the mitochondria have two membranes, an outer one, which is seen here in purple, and then you've got the inner one, which is here in like a light purple. These membranes are made out of phospholipids, just like the cell membrane has phospholipids, and the outer membrane covers the surface of the mitochondria, while the inner one has many folds, and these folds are important not only for surface area, but in there, the folds we call crista is where the action takes place. So the folds increase the surface area of the membrane, and that's important because that's where all the protein are held, and there are many protein molecules that are involved in the electron transport chain, and this is where energy is produced. It's also where a whole lot of other chemical reactions that I mentioned earlier that the mitochondria is also involved in. So if you look at it, you're sitting with the outer membrane, you're sitting with the inner space, you're sitting with the crista, that's the folds. The inner membrane is where all the chemical magic is happening. You've got the ribosomes sitting in the center, and basically you got some DNA material as well, which we're going to touch on in a little bit. So let's look at the genetics of the mitochondria. So here I had my own slide, but we're finding it difficult to incorporate it into this structure. But if you look at the human cell, we've got about 20 to 21,000 genes, and they basically produce, some of them are active, some of them are inactive, and they there to code a whole lot of protein molecules. So if you look at mitochondrial genetics, there's over 1,100 to 1,400 distinct proteins that are involved with the mitochondria. Most of them are produced by nuclear DNA material, but are necessary for the mitochondria to do its function. And then the mitochondria itself has the ability to produce some of its own protein, and that's because it's got some genes which we're going to cover in a little bit. Now, mitochondria, if you look to the right, actually comes from the maternal cell line, and it's very interesting that certain religious groups, well, only your mother can tell who your father was, and it's, of course, it's because of this interesting pathway. But it is this DNA material that you find in mitochondria that's allowing people to trace where your origins are in terms of, according to the first African gene that moved out, and you can trace it back to very few mothers, historically speaking. So let's show you where the mitochondria comes from. So if you look at the egg cell, it has the nucleus, and that means it's got nuclear DNA material. And then you've got a whole lot of mitochondria, and they have their own DNA. So this is in the egg. In the sperm, in the head of the sperm, you're going to get nuclear DNA material. And then in the tail, you get the mitochondria, so that it can actually reach the egg for fertilization, because that's the guy that needs to beat and it needed energy. Now, once you have fertilization, remember it's only the head that manages to penetrate this part of the egg. And so when you look at it, the nuclear material comes from both a sperm and egg, whereas the mitochondrial one only comes from the mum, because that mitochondria in the tail is lost. So all mitochondrial DNA is inherited from mum, and that becomes important later for some of the inherited conditions. And this is important. I'm going to reiterate it again, that nuclear genes are important for mitochondrial function because they produce the majority of the proteins that are going to be used by the mitochondria. And obviously here you get maternal and paternal genes coming through. So you can have defects coming from both mum and dad from a nuclear DNA perspective, but from a mitochondrial perspective from mum, it mainly comes from maternal inheritance. And so you're going to see that playing out in a few medical conditions that I made a list for, and I'm going to try and import that part of the talk a bit later. So let's look at the mitochondria in health and disease. So healthy mitochondria are directly linked to wellness, whereas dysfunctional mitochondria present with disease. And we see this at its most extreme in inherited mitochondriopathies. So mitochondrial dysfunction is implicated in a spectrum of human diseases, with dysfunction being the core feature of virtually all non-communicable chronic diseases. Now, certainly that wasn't known when I qualified. We used to mainly look at primary mitochondriopathies, and they were mainly in the neurological wards, and every now and then you saw an odd case or you heard the consultant speak about it. So mitochondrial dysfunction underlies greater than 50 inborn errors of metabolism, and that list is growing. Type 2 diabetes, neurodegenerative diseases, and cancer are among some of the other conditions that will affect mitochondrial dysfunction, and mitochondrial dysfunction indirectly affects them as well. So it's actually an incestuous relationship. Mitochondrial impairment can be inherited, and it can be inherited through autosomes or X chromosomes, which are are essentially maternal. Or they may develop in a multifactorial way, including and not restricted only to toxic environments. So you've got the inherited forms and basically the acquired forms. And that's what we're going to be touching on a bit later. So mitochondrial dysfunction is associated with accelerating aging, neurodegenerative conditions, tumorigenesis, metabolic syndrome, mood disorders, amongst other medical conditions. So can you see why it's important to know stuff about mitochondria and how to test it and influence it? Because there isn't a disease process that we currently deal with that doesn't have one way or the other the possibility of mitochondrial dysfunction. Mitochondrial injury and subsequent disturbed energy homeostasis and uncontrolled ROS production can contribute towards neurodegenerative conditions. We mentioned malignant cell transformation and cardiovascular disease. So although disease origin is common, its presentation in individuals may differ. So one person with type 2 diabetes may have different manifestations in different organs from having abnormal mitochondrial functions, and in the heart, the mitochondrial function may be a bit different to the muscle or the pancreas. Dysfunction may occur across one or multiple mitochondrial function outputs, and we'll show you a little bit of that in the later modules. Now, let's see what's happening. So primary versus secondary mitochondrial disease. We use primary mainly in the past from a genetic perspective, inherited perspective, and secondary from acquired. But you'll see here it's a bit different because you can get acquired mutations later in life, and that's clumped under secondary as well. So primary mitochondrial disease are mitochondrial diseases that are caused by a germline mutation in the mitochondrial DNA or the nuclear DNA. And it happens in the gene that codes for either the OX4 structural proteins or mitochondrial proteins of a complex machinery that you're going to see, and there's about 6 of them, and we're going to touch on that a bit later. The secondary mitochondrial disorders stem from gene defects outside of the OX4 system, and these come from later life insults. So anything that's going to disturb the mitochondrial function from a control pathway or trigger oxidative stress can cause these later life insults. These disruptions can arise from thyroid disease, from diabetes, from cancer, from aging, neurodegenerative conditions. And so we've all had the thyroid patient where you've sorted the TSH out, you've given them enough thyroid hormone T4 or T3 or a combination, and they'll still complain of fatigue. You look at the adrenal gland, you sort out the adrenal gland, there's no iron issues, and they'll still be fatigued. And you can see why the mitochondria will then play an important role. So unlike primary mitochondrial disease, which are always inherited, secondary can be inherited or acquired through basically later life insults. So the way that slide ended, and I went through it quickly, but it's quite verbose, is in some conditions, just from signs and symptoms, it's very easy to distinguish primary from secondary causes, whereas with mitochondriopathies, that's not always the case. So certainly in some of the inherited syndromes, because you've got a cluster of conditions, you know that that will be one of the primary mutated conditions, but it's not always the case. So mitochondria are found everywhere except red blood cells and impact virtually every organ system. And I think that's the message I need to relay today, that it doesn't only belong to the neurologists. So they do more than energy production. I spoke about its importance in other factors as well. It's impacted by inherited issues, some internal factors, and external factors. Internal factors would be like hypothyroidism, diabetes with an increased production of free radicals and so forth, whereas external factors would be maybe pesticides, for instance, or heavy metals, or drugs that you and I use on a day-to-day basis. The mitochondria play a vitally important role in the aging process. It's an integral component of cell signaling in terms of one cell talking to the other, and then from a survival perspective as well, from a cell perspective. And we spoke about apoptosis there as well. And you'll find it in basically every organ system. And the beauty of this is at the moment, with your blood, you can send off a general and you can get a general idea in terms of what the mitochondrial function is, and we'll show you in the second talk an example of what kind of report you can get when you send the test off. I just sent off, I think, 13 or 16 samples last week, and looking forward to the results in the next 3 weeks, and so we'll see the correlation between the mitochondria and these patients that I've assessed on. But basically, in the future, what they're looking at is they're going to, this particular university is already working on it, where they will able to localize without a biopsy thyroid mitochondrial pathology as opposed to heart, which is important in cardiac failure, in fatty liver and other liver diseases, kidney issues. So that will take it to the next level. And so we're all excited about what's coming. So summary of the first module: mitochondria are organelles that are maternally passed along from individual to individual and contain unique set of genes which, along with nuclear genes, express genetic material in a way that is unique to that particular individual. The mitochondrial structures are unique and play an important role in how the mitochondria functions and key processes. Mitochondria do more than just produce energy, and they are linked to disease and health. And so if anybody wants references, just speak to Leonie and she'll send it through. So let's go on to module 2 now. And here we're going to be looking at biochemistry and physiology. We're going to try and keep it simple. So you'll see we're trying to string this 4 modules to show you where various products were able to be used to have an influence on improving mitochondrial function. And that's why it's important for us to understand each aspect of the mitochondria. Whereas when you and I are trying to manage patients with type 2 diabetes, unless you're an endocrinologist or a anesthetist, you didn't really need to know every bit of functioning of the beta cell at a biochemistry second messenger system level. Whereas here, you'll see why it's important, because you can see where your product is going to be working and why it needs to be individualized. So here, the learning object in module 2 is going to be describing the physiological and biochemical processes associated with the mitochondria. To understand these processes, compare normal with abnormal physiological and biochemical processes. So let's start, and there's very few slides to module 2. So the main thing to understand from an ATP metabolism perspective is the oxidative phosphorylation pathway. So the OXPHOS pathway, which is shortly known, is a biochemical process that occurs in the inner membrane of the mitochondria, and this is where energy, ATP, is produced. It is central to cellular metabolism and generates ATP, plays a critical role in maintaining cellular homeostasis, and it's important because this is where you need to be regulating the reactive oxygen species, which we're going to talk about as ROS. And it supports various other metabolic pathways. So it involves the electron transport chain, which is a series of protein complexes, and there are a total of 6, and these are all basically coded for in the mitochondria, and associated molecules that transfer electrons from reduced substances like NADH. And later in my other presentation, I've got a bit more information on that, and FADH2 to molecular oxygen. So it's moving from the electron transfer chain, using oxygen and generating ATP. This electron transfer is coupled with pumping of protons across the inner membrane, trying to create an electrochemical gradient known as the proton motive force. So it's no different to a battery, because that's exactly what the battery is doing, trying to create a gradient so you can have transfer of the energy molecule from one substance to the other. So if you've got a lithium battery, it's going to be basically lithium. They've got a hydrogen fuel cell battery, it's going to involve platinum or one of the heavy metals and oxygen. And you can get sodium batteries as well, and so forth. So it's basically to try and create a gradient so you can have a movement of hydrogen molecules to create energy. So the energy is stored in this gradient and then drives the ATP synthesis via an enzyme called the ATP synthase, complex 5. So we liken it to basically every cell in our body has a battery, and this is the same process here. Oxphos is a major source of ROS, so that's the problem. So just like you got a combustion engine in a car, and you need to combust oxygen with petrol to generate energy to drive the pistons to move at the end of the wheels. In that process, there's a lot of waste being produced, and that waste can become sort of toxic to the car. And so if you don't service your car, you don't change your radiator fluid, you don't change your oil, you don't change your petrol filter, eventually that car is going to deteriorate. And it's the same thing here. Humans need to survive. We need to breathe. And part of breathing in oxygen through a series of chemical reactions, the waste product are many, but we also happen to be generating reactive oxygen species. They will then steal electrons from the cell that is sitting next to it and basically cause disruption. And what we humans call aging is just basically ROS accumulating or destroying certain organs. So when that happens to a heart, eventually it's cardiac failure. When that happens to the endothelium, it's eventually hypertension. When that happens to the brain, we give it a name and we call it dementia or Alzheimer's and so forth. So this is the underlying problem with aging. And you may say, but you know, what differentiates the system in a young person and an older person? The engine starts getting older. And so the more ROS you start accumulating as you get older, the more damage there is. And that is why you'll see a kid who gets cut will heal much quicker than somebody my age. It's one of the processes. Of course, the hormones play an important role, but yeah, it's basically free radicals that causes damage. So during the electron transfer, a small percentage of electrons leak from this ETC system and then reacts with oxygen to form superoxide anions, and that becomes dangerous. And so it's not a perfect system. There is leakage into the system. These ROS can further convert into hydrogen peroxide and hydroxyl radical groups, which are highly reactive and can damage cell components, whether it's DNA, protein, or lipids. So when you start affecting the lipid layer on the cell membrane, there's going to be leakage and there's going to be a problem, and you're going to influence influx. When you start damaging protein, you're starting to damage second messenger systems. And of course, proteins get incorporated into many other things as well. And then once you start messing with the DNA and you cannot clean up your DNA mistakes too quickly in terms of the mutations, these mutations are then acquired through life. That's what we call later life alterations. And then we're going to have damage and aging. So mitochondria have evolved antioxidant systems precisely to try and counteract that. And of course, when that deteriorates, there's trouble. So what kind of an antioxidant system is in play? It's the superoxide dismutase, the SOD system, and glutathione peroxidase to mitigate this ROS toxicity. That is why when somebody has a Panado overdose in a hospital, and I mean, we're talking about like lots of Panado, that person's going to end up dying of liver failure unless you infuse high levels of N-acetylcysteine, and N-acetylcysteine will directly influence the production of glutathione, and then glutathione mops up the free radicals and basically ends up saving that person's liver. And so that is what you see glutathione do in real life in the wards when you're training, but this is what it's doing on a daily basis at a cellular level. Oh shit. Okay. That's why I had that side thing opened up so that I can cancel this function every now and then. So where are we? Okay, so mitochondria have evolved, so we are on the bottom to have that antioxidant system, and then the oxphosphorylation pathway is closely linked to mitochondrial biogenesis. And that's important because, and this is very important because this shows that you can regenerate mitochondria. And so your cells are producing mitochondria all the time. And that's why if I lead a sedentary lifestyle and then I start doing what Bruce Fordyce does, I'm going to be able to influence my mitochondrial density. Now let's look at the mitochondrial proteins that I mentioned earlier. So this is the DNA material that is only found in the mitochondria that codes for a whole lot of protein. And I think the mitochondria has 33 proteins. I've got that as a slide, and we're going to go through that. And basically, it codes for in and amongst these protein a few other. So complex 1, 2, 3, 4, 5. There's 6 of them, but the 5 are the main ones that are involved in ETC, electron transfer chain, and located on the inner membrane of the mitochondria. The 2 electron carriers, NADH and FADH2, begin the chain by donating the electrons to complex 1 and 2 respectively. These electrons are then passed along the next complex in the chain. And so I'm going to show you a diagram later where the Krebs cycle, where the thing gets passed on in terms of generating ATP. ATP synthase allows the proton motive force that I spoke about on the last slide to be discharged and utilized by the cell. This energy generated by hydrogen ions diffusing back into the matrix from the inner membrane, remember the middle part between the inner and outer membrane, happens via complex 5, and this is how ATP and ADP is connected. Now this all becomes important later on the last module, where we show you how each of the different molecules that you could be using, natural or synthetic, will influence these various pathways. The electrons, meanwhile, combine with hydrogen ions, oxygen to form water, and this is done by complex 4. This process is not perfect, and I told you some electrons will leak out and basically produce ROS, and then react with oxygen and cause free radicals, and that's what's causing trouble. So let's move on to the importance of the bilayer. I'm going to digress a little bit here because in one of my previous webinars, we spoke about oils and how the right oils are important to form the right membrane around a cell. So one cell needs to communicate with the other. The cell needs to be able to have access to a whole lot of nutrients, trace elements, vitamins, cofactors, and somehow they need to get into the cytoplasm. And that happens because of the magic of this bilayer. It has many pores that will allow things to come in and out. And the fluidity, as opposed to the rigidity of this membrane, has been linked to many disease processes. And that is why the balance between omega-3, 6, and 9 becomes very important. And so we've moved away as modern society from where the cavemen used to have a ratio of these oils that I mentioned in my last webinar. We are now sitting with ratios of between 20 and 30 to 1, going in the wrong direction, and that affects the rigidity. And just to give you one example, that is the cause of insulin resistance, right? That is why that signaling pathway goes wrong. So This cardiolipin is very important. So phospholipids are the main building blocks for the mitochondrial membrane, inner and outer. Cardiolipin is a unique phospholipid localized and synthesized in the inner membrane. It is now widely accepted that cardiolipin plays a central role in many reactions and processes involving in mitochondrial function. The signature mitochondrial lipid is this cardiolipin. It is a lipid dimer with two phosphatidyl groups connected by the glycerol block backbone. This was first isolated, long time ago, about 80 years ago, and is shown to reside nearly exclusively in the mitochondria. It constitutes about 20% of the entire total lipid mass of the inner membrane, this cardiolipin, and it's essential for various mitochondrial processes, including the fission and fusion process. It's important for the OX-phos complex, and it's important for the supercomplex assembly, which we're going to talk about in the next slide, and the crystal structures. Cardiolipin plays an important role in the mitochondrial membrane morphology, its stability, dynamics, mitochondrial biogenesis, making new ones, the importation of protein from outside of the mitochondria to inside the mitochondria, mitophagy, very important role in functional medicine and aging, and basically all the different pathways involved in mitochondrial involvement with apoptosis. So alterations occurring in the cardiolipin profile may negatively impact the activity of various proteins that are involved in the mitochondria, whereas enzymes that are involved in mitochondrial function. And it goes beyond just ETC and OX-phos complexes, but these are the main ones. These events may play a causative role in the etiology and progression of several pathophysiological situations and diseases. One that is known, which we've known for a long time, but I told you that list has expanded, is like the Barth syndrome, mitochondrial ischemia, reperfusion injuries, heart failure, diabetes, neurodegenerative disorders. So several agents, and I'll give you a list at the end from other part of the talk, will directly or indirectly target cardiolipin, including fatty acids like phosphatidylcholine, elamipretide, melatonin, plastoquinones, derivatives, ubiquinols. They have all been proven to preserve the integrity of cardiolipin. And so it's important for mitochondrial function and treating disease or disorders related to the mitochondria, and you're going to see that in the 4th module. Now, some of the other processes that go beyond the OX-phos, the production of free radicals, apoptosis, and the ETC transfer, is this process here. This is the ER, endoplasmic reticulum, mitochondrial encounter structure, so it's called MS. Because remember, when you're looking at the cell, always near the mitochondria, you'll find the endoplasmic reticulum. And that's because there's an interplay between the ribosomes and some of these substrates, and we're going to go to it. So the EMS complex, which serves to tether the endoplasmic reticulum to the mitochondria, spans between these two. And so from the ER membrane to the mitochondrial outer membrane, that space is where the magic happens from a point of view of energy production, protein and substrate transfer. So substrates and products must transit between these two organelles for an efficient phospholipid biogenesis, and mutations that will disrupt this complex will have end results and consequences. So it basically slows the slows the processes down, never stops it completely. And so basically you just have a slower battery. The conversion of phosphatidylserine to phosphatidylcholine is also then required for this transition to happen. The connections are mediators in mitochondrial fission, and it happens between ER and mitochondrial contact. We said the mitochondria is involved in other processes as well. So calcium signaling becomes very important and plays an important role in almost every aspect of cell function. One cell needs to talk to the other, and hormones need to do their work in the cells, and they do it through signaling pathways, and the calcium signaling pathway is one of them. So the rapid, transient, and high amplitude alterations in calcium concentrations are critical for hormonal release. They're critical for transcriptional regulation, important for cell death initiation, and many other processes. The MCU1 protein is essential for mitochondrial calcium uptake, and you find that in the membranes. And the role for the MCU, which we're going to go through a bit later, and the MCU is this: mitochondrial calcium unipotter, have already been established in insulin secretion, neuronal excitation, that means sending a message from one end of the body to the other, cell death, and other systems. Another system that's important is the mitochondrial pyruvate carrier. Pyruvate is an essential component in the Krebs cycle to produce energy. So pyruvate transports into the mitochondria. It is then oxidized to enable it to produce ATP. And this is the most common fate in all cells of the body. Several diseases are characterized by a decrease in pyruvate oxidation. And you find this in a lot of cancers, you find this in heart disease, and in the old-fashioned classic inborn errors of metabolism that we know, some are acquired and some are inherited. Okay, I've done something it doesn't like. There we go. So in summary, and mind the spelling on this guy's slide, but biochemical processes within the mitochondria play a critical role in maintaining energy production, cellular homeostasis, regulating ROS, and then supporting various other metabolic pathways that I've just highlighted on the last slide. The moving of electrons efficiently along the electron transport chain is critical. And when degradation occurs, there's mitochondrial dysfunction. And this is going to result in biomarkers that we're going to be able to read when we send our clients tests off. Primary and secondary diseases cause degradation along different parts of the electron transfer chain. And understanding with specificity where the degradations are occurring will result in an impact. And that's where the biomarkers are going to be important for you to be making certain decisions. Biomarkers associated with the mitochondrial biochemistry are impacted by toxic stresses and normal day-to-day influences, which limit the ability to utilize single markers or tests for mitochondrial dysfunction. So that's why this new test becomes important. So I'm not going to go to module 3 today, but we're going to jump onto, and let me see if I can pick up my other slide that I wanted earlier. No. There you go. See, they just send the educational deck 11 minutes into our talk for me to be able to change it. Unfortunately, it came a bit too late. Because I wanted to incorporate these slides that I'm going to show you now into the talk so it becomes a bit more meaningful. Can you guys see this?
No, we can't, Dr. Barner.
Okay, I'll share it. I think I've been talking to myself for a while, but it's fine. I do that quite often. Can you see it now? Hey, go on.
Still not.
Yeah, sorry, I just lost it there. Now, Leonie, can you see it?
No, unfortunately not.
Okay. Let me just stop sharing all of this. I'm going to just go out and come back. It's a pity, half an hour earlier and we would have able to incorporate all of this. Yeah, this video. I'm sure you can see it now.
Yes, I've got it.
All right.
Perfect.
Okay, let me see if I can. Okay, so this was just to show you the mitochondria and the relationship to the endoplasmic reticulum and the space between it. I mean, we were never taught that in biology, but there's a close proximity because this guy and this guy forms a complex. So the outer membrane there and the outer membrane here, and basically a lot of the transfer or chemical processes happens in between that space. So we get a better understanding. So the two key molecules in terms of energy production in the Krebs cycle, and there's many, but NADH. So I've lost the marker. Okay, but I'm giving it away. So it's nicotinamide adenine dinucleotide. Those of you who do functional medicine, you know the big story about NAD, and this is where NAD comes in. And so if you look, NAD is reduced from NAD plus in the oxidized form, and that cycle basically is important for basically electron carrier from one part of the Krebs cycle to the other. And then the FADH actually stands for flavin, which comes from riboflavin, which is vitamin B2. So I find, I mean, look, I've been teaching for years now, and a lot of the youngsters, and maybe they get it from the older teachers, don't give too much attention to the importance of trace elements, cofactors, and vitamins. But let me give you an example. I mean, I remember when I qualified in our exam, we actually, as a student exam, we actually got a case of beriberi. And you don't see beriberi now because thymine is fortified and it's found in many foods. But in my day and age, you used to every now and then get patients in just cardiac failure and they're severely sick. And it's literally like magic. They get admitted close to the doors of heaven. They're almost going. You give them an infusion of thymine and jet fuel, and like within a day or two, it's like magic. They're completely out of cardiac failure and they sort it. That's the magic of thymine. And, you know, we pay lip service to say, oh, it's just expensive urine. But that's a classic example of how narrow the window can be from life and death in the extreme situation when you have absolutely no thymine. And then there are many other medical examples. I mean, we hardly ever see that now, but in the old days with when patients came in with severe alcoholic intoxication, you would have what we call the drawing back neuropathy. So just on the chest, the nerves come along right up to the sternum, of course, under your skin. And what happens is those nerves start dying out and they lose sensation in the middle of the chest. And I remember we had to take a needle. And if you didn't do this in exam, you would have failed because you had to do it for all the patients based on the history in terms of why they came in. And then you would able to demonstrate that the patient has lost nerve function. So now we do it with diabetes and peripheral neuropathy, but with alcoholism, we used to see it classic here. Now remember the alcohols are fortified, so that's why you don't see it. And it was interesting because as a student, as you were in the ward, they came in, they had neuropathy elsewhere as well, but it was nice to examine for this one because as you gave them jet fuel and you gave them the vitamin Bs, the nerve function improves, and every day when you test, you can see the nerve function improving because they can feel it closer and closer coming to the chest, and eventually that nerve heals. So I digressed a bit because you're going to be seeing on my next few slides the importance of these nutrients. So both of these, you'll see on the bottom, it says both NADH and FADH2 are essential for aerobic respiration and production of ATP. So I'm discovering ChatGPT now. I've got level 4 paid for version because my son's in university and I'm doing a course on how to prompt these things to do. So bear with me. This was my first exercise on Jasper AI and I think Dell E. And so here's a Krebs cycle. Now, if only I can get a, where's that intelligent person who knew how to use this computer earlier? Because my son's not here. I'm looking for that bloody pointer. Okay, leave the pointer. So here's the Krebs cycle. And you'll see on the bottom, there's the FAD. You get succinate. Then you get NAD. That gets reduced to NAD plus with water will become oxaloacetate. And that then becomes citrate. And then alpha isocitrate, alpha glutarate, succinyl. CoA will then play on it and become succinate, fumarate, and then you've got GDP, and then you've got ATP. Now, there's the cofactors that are important in the pathway in color. So without those cofactors or a shortage anywhere here, you're going to have an arrest in the cycle, and you're going to not have your 36 ATP molecules coming out of this cycle. You're going to move into aerobic. Anaerobic is respiration and just produce 2. So it's not an energy efficient cycle. Now let's add where some of the vitamins will work, with thiamine, niacin. Let's look at some of the other products like calcium. You can see copper on the bottom. You can see iron. You can see the important role of magnesium. We had a whole talk on magnesium and we spoke about how it's important for 400 different functions in the body. Now add some trace elements, vitamins and cofactors and minerals and put them together. Can you see how complicated this pathway is becoming? So if you're short of 1 or 2 of any one of these essential factors, you can have an arrest in the basically Krebs cycle. And then you're sitting in trouble from an energy perspective, right? And so from that perspective, we pay lip service to these things, but they're very important. And the last slide shows you where ATP comes out in yellow on this particular Krebs cycle. So it's a basic attempt because, you know, you can get the ones off the net becomes become so complicated. And those of you that heard me speak on obesity, I show you this slide with the biochemistry, and there's so many different pathways, but all important. So yeah, so this was to show you that the human nucleus has 20 to 21,000 genes. They are basically important for coding protein, but you get non-coding genes as well. For RNA, you get the transfer RNA, you get the ribosomal RNA, you get the mRNA, and then they're important in the regulatory sequences. So you got the promoter gene in front of the gene you're going to code, and then the encoding and the enhancers. You got the repetitive DNA elements, and so the entire human genome has over 3 billion base pairs. And so from that perspective, it is not only about the production, if you look on the bottom, in terms of the proteins that come out from the 20,000 genes, but the functional capacity, the ability to regulate genes, splice them, interaction becomes important. So here's the mitochondrial DNA. There's 37 genes, and what do? 13 of them code for the complexes that I mentioned earlier and the ATP synthase enzyme. 22 of them code for transfer RNA, and that's important because remember we spoke about the mitochondria and the ER complex and what happens in between. This is where the transfer RNA comes in and protein synthesis, and then 2 of the genes code for ribosomal RNA, and that's important as well. So the mitochondrial RNA is circular in pattern. It's inherited maternally. It's located in the mitochondrial matrix. I think I showed you that in the first cartoon. Replicates independently to the nuclear DNA. That's important. And most mitochondrial proteins that are required for the mitochondria to work are actually coded for by the nuclear DNA and then imported to the mitochondria. So the mitochondria make those from the 33 genes that I showed you, but there's a whole lot of other ones that come from nuclear DNA. That means you can have defect from mother and father in that nuclear DNA that is inherited, because that means there isn't going to be 1, 2, or 4 of the proteins that are required for the mitochondria to work. You could get inherent X-linked maternal mutations coming through and affect mainly the mitochondrial protein. And then you could have outside factors that can independently, through a non-genetic transfer pathway, affect mutation directly to the mitochondria and directly to the DNA material that's coding for the protein that the mitochondria are going to need. And so both these DNAs are important. Now, I'm just going to quickly go through this because for some of the doctors in the audience, it may tweak what we learned, and we never saw some of these cases because they're very rare. And so you had the Leber hereditary optic neuropathy. Now, this one is easy because you know it's inherited. It's from the maternal mitochondria. And these patients have sudden painless central vision loss in young adults, mainly males. You got the Mella syndrome. I remember being asked about that by Prof. Modi in one of my tutorials. Here you got mitochondrial encephalopathy, you got lactic acid and stroke-like episodes. And you can look at the mode of inheritance. We've heard of the MERVs, where the ragged red fibers, so you do a biopsy and you'll see this. These patients have epilepsy and myoclonus. You have the Lee syndrome, you've got the Kern-Sayre syndrome. Now, we thought the importance of mitochondria during certainly my studies was limited to these 5 conditions. And so we said, oh, what the hell, we don't really need to know this, because when the hell am I going to see a Kern-Sayre? And even if I see a Kern-Sayre, what the hell am I going to be able to do about it? Now it's different. So there's a whole lot of secondary mitochondriopathies, and we spoke about these, and these happen because there's damage to the cell or to the nuclear DNA material or the nuclear mitochondrial DNA material. And so if you look at some of the common causes of secondary mitochondriopathies, you get the ones that are genetic related, like Friedreich's ataxia or Wilson's disease, where you have an accumulation of copper and that becomes toxic to the mitochondria. You can get the liposomal storage disorders like Gaucher's, the Niemann-Pick syndromes. So those are still genetic, but acquired through either heavy metal poisoning or some of the other mutations. Then you can get metabolic disorders. So diabetes. I mean, there's over 750 million people worldwide sitting with diabetes, and then a whole lot of people with pre-diabetes. So this chronic hypoglycemia is an oxidative stress. Make no mistake, there's damage to the mitochondria, whether it's in the mitochondria of the nerves, kidneys, eyes. Hypothyroidism affect mitochondria as well, and so both in number and function. What about some of the environmental factors that can lead to mitochondrial pathology? Alcohol abuse. It will lead to mitochondrial membrane damage and impair the oxidative phosphorylation pathway. Certain drugs. So certainly in the early parts of HIV treatment, we saw how toxic these drugs were because they affect the mitochondria. Valproic acid. Some of the antibiotics. Some of the chemotherapeutic drugs, definitely the ones related that are derived from platinum, cisplatinum. Pesticides, heavy metals, lead, mercury. Some of these things were in your fillings. Some of these things were in the paints. The lead pipes that we used to transport water from place to place. Only now we're using the polycop plastic pipes. So whether you're looking at age-related mitochondrial decline, whether you're looking at some of the chronic metabolic conditions that I've just mentioned, whether it's some of the inherited ones, you and I are going to be able to now change the lives of some of our patients, because even patients with neurodegenerative conditions, sarcopenia, there's a role to be played here. And in the future, we're going to be able to even go and do organ-specific mitochondrial tests. So, yeah, I've mentioned this, so I'm going to leave it there. So these are just some of the slides I wanted to fit in where the slides matters and digress and expand on it a bit. But I'm going to stop here. There's 2 more modules. And by the end of the 4th module, you'll be able to fully understand how the mitochondria work, we hope. And then I'm going to give you an example of what a result looks like. And you can see there's basically the report comes in, gives you 4 major sections, and each section has 3 different layers to it. And I'll give you an example of how I look at the first layer. And I'm only using this example not because I love cars, because my child loves cars, and then I started learning and understanding a bit more about cars. But if you had to look at a car that is idling, while the car is idling, it's still using up some petrol to idle and to stay switched on. So your mitochondria can do that. They have to be idling all the time for that cell to use up energy. And then you start normal function and you need to upregulate that. So the ability to go from a point where the car is just stationary, but the car is switched on, to acceleration, you need certain processes to happen to the mitochondria to upregulate their function. And then if you want to do with a Mustang a 1/4 mile and you want to do it in less than 6 seconds and you need that burst, then there's a different part of the mitochondria that comes into play. So when you're looking at clients with fatigue and you've excluded anemia or thyroid, that's when you'll have a better understanding in terms of where that pathology lies. Or when you got in an adrenal gland and the patient's getting energy dips, you're able to understand why along that pathway it's leading to symptoms X, Y, and Z. And so I don't know if I'm at liberty to tell you guys this, or Benji will tell you guys at some point, but Benji is a doctor and he is involved in the distribution of the mitochondrial kits. The tests are 7,500 rands, and the doctor gets to keep a certain portion. I think it's about 10% for basically doing the test, interpreting it, and then of course offering the client. So it's just like the genetic test that we do in South Africa with some of the other companies. And so it's exciting times. And there's videos that show you how to collect the bloods, put it in the plastic, and basically send it off. Once it gets dry, it gets over there, and then I've done, I told you, a podcast with the doctor that's been responsible for inventing or discovering this test. And he speaks with lots of passion in a nice overview in terms of the importance of mitochondria, where they're going, and how you can use it in your practice. But Benji is going to be distributing that more widely in the near future. So I'm going to leave it here. I'm going to see if there's any questions. And you can put the questions in the chat. And basically anybody can order this test. You can be a pharmacist, you can be a physio dealing with patients with a lot of fatigue, an osteopath, homeopath, functional medicine doctor, cardiologist, neurologist. Patients can order it themselves. The test has been widely distributed from, I think, the second quarter. It's relatively new in the world, and basically it's going worldwide. You will get CPD points for this and for the next one, because I see somebody asked for the CPD certificates. And no, we can't see anything. How useful is NAD? Okay, Dr. Ghani, don't get too excited. We're going to be talking about treatment and the 12 different products that can be used. But first, you need to know where we're going, why the mitochondria is important, that it goes beyond just the 5 inherited primary mitochondriopathies that I was taught at a world-class medical school. And you can see how it's expanded. And then we've gone through the biochemistry, and I went through it slowly because, yes, some of the words were complicated, but it shows you the various different pathways that are involved. And so then when you do the test, you'll be able to see that in this patient, it's likely that you need to use product A, B, and C, whereas in another person, it may be D, and in another person, it may be a few other products. So we're going to get there. And yeah, I see part 3 and 4 is on the 7th of August. And she says there's no need to re-register, which is a good thing. And if there's no other questions, I'll leave it there. I'm going to get the new kit now, because you can see he just sent it through me at 11:10. So then I will able to modify it according to the slides. So then I don't have to scroll down, because it does affect the attention. And I'll put in some of the basic stuff that we used to learn in BSc and in medicine, and to make it a bit more relevant to your practice and my practice. So thank you for your attention, and sorry for a few glitches. But yeah, we have managed to get a non-PDF copy now of the modules, and then we'll take it from there. Good night, and thanks for your attention.